we've got two candidates for perhaps the earliest hominin in the fossil record. And both of these date to roughly 6 million years ago. We refer to these and then later Ardipithecus as pre-Australopiths. So they date, range from about 6 plus to 4.4 million years ago. The first of these is Sahelanthropus chidensis. The first of these is Sahelanthropus chidensis, who dates to between 6 and 7 million years ago and was found in northern Chad. Now, Sahelanthropus is also known from one single skull that was dubbed Tumai, and initial dating hypothesized closer to the 7 million year mark, but more recent analyses have come back closer probably to the 6 million year mark. Sahelanthropus chidensis has a number of unusual characteristics, including a small brain case, roughly chimpanzee sized, a robust skull that has a sagittal crest and pronounced brow ridges. Both of these, which we talked about and will continue to talk about, are associated with the massive musculature um, required by a diet of hard chewing, a smallish vertical face with front teeth that look like later hominins, non-honing canines, reduced canines, and a foramen magnum that is intermediate between chimpanzees and humans. So we're seeing this transition process I mean, we can't definitively say that Sahelanthropus chidensis is a biped, given that we don't really have postcranial elements, we only have skulls. But the movement of the foramen magnum indicates at the very least significant time spent upright. The second of these really early hominins is Auroran tutinensis, who dates to about 6 million years ago. We've got skulls of Auroran found in both Kenya and Ethiopia. Most of these are dental remains. We do have some lower limb bones. Those lower limb bones are much more like later hominins, which then indicate bipedality as a locomotor movement. Then a little bit later, about 5.8 to 5.2 million years, and then another species at 4.4 million years, we see the genus Ardipithecus. Ardipithecus was no doubt fully bipedal. The Ardipithecus fossils that we found so far are found in Ethiopia, and the climate of this environment that Ardipithecus evolved in is mixed woodland. It's not dense tropical rainforest. It's not grassland. It's kind of this mixed wood with clearing areas that's really similar to what some extant populations of chimpanzees live in. This is what gives us the indication that bipedality first evolved in the forest. That bipedality didn't come after we moved out into grasslands. And so this factors into determining the whys of bipedality. What was it that favored it evolutionarily? And it was likely these energy savings that we talked about with part two of this lecture. So Ardipithecus, similar to Sahelanthropus, has a chimpanzee-sized brain, has a height that's closer to 4 feet, and a body weight around 110 pounds, has a short and broad ilium. So we're seeing shifts in the pelvis. That foot has lost the grasping big toe, though we don't yet have the appearance of the arch. When I say lost the grasping big toe, it's not completely gone, but it's migrating. It's moving more in line with the other toes. The limb proportions, the pelvis, the length of the arms, in particular long curving fingers, indicate that Ardipithecus was still quite an adept climber. And in a climate that's a mixed woodland kind of habitat, that makes sense. You're going to see significantly more time spent on the ground, but you're still going to rely on resources that are up in the trees. You are also going to uh, need to climb trees in case of predation or for safety at night. And so here we have some pictures that show some of these changes. We've got the skull tumai in the top left that's associated with Sahelanthropus. We've got a comparison of the foramen magnum between chimp skulls, modern human skulls, and Sahelanthropus. So we're seeing the foramen magnum Migrating forward, we see then the skull and the postcranial elements of Aurora tugenensis. Noticeably, we've got this bulbous protuberance on the top of the femur that's shifting towards more of an orientation that we see with upright bipeds. And then Ardipithecus, we have almost a complete skeleton of Ardipithecus. It's lovingly referred to as Artie, that was found in the early 2000s, that really does demonstrate this commitment to bipedality. Following the pre-Australopiths, we have the emergence of Australopithecus as a genus and the Australopiths as a group as a whole. And the Australopiths persisted for quite a long time evolutionarily. We see the earliest Australopiths appearing around 4.2 million years ago. We find an abundance of Australopiths after about the 4 million year mark, persisting all the way until about 1.2 million years ago. The Australopiths are comprised of two closely related genera, Australopithecus and Paranthropus. The early specimens are grouped with the Australopithecus genus. The later specimens see a specialization between what we call gracile and robust Australopiths, with the robust Australopiths falling under this genus Paranthropus. Australopiths are found across all major regions of Africa, from South Africa to Northern Africa, east to west, a variety of different climates. They persist for more than 3 million years, so they are arguably pretty successful. They've got uh, considerable complexity in terms of evolutionary diversity, and all of the Australopiths share some major features. All are very clearly bipedal. All have relatively small brains that are not much larger than chimpanzees. We see a slight enlarging of the brain case, but that's associated with slightly larger body size as well. And all of the Australopiths, even if they were gracile Australopiths, which we'll talk about that distinction here in a sec, all have large teeth, particularly the back teeth. And those teeth are very, very heavily enameled. And so we see teeth changing, like we discussed with Darwin's finches, that we see the thickening of enamel and changing tooth morphology evolve pretty rapidly, in response to the shifting diet that comes with more time spent on the savannah. So Ardipithecus demonstrates that bipedality evolved in the forest, but Australopithecus, or the Australopiths as a whole, demonstrate pretty successful life on the savannah. They're fully committed to this different ecological niche that's characterized by the patchiness of these resources and this thick, fibrous vegetative material. The early Australopiths represent perhaps those that could be ancestral to the genus Homo. We've got at 4.2 million years Australopithecus anamensis. This is a potential early ancestor for both the later Australopiths and the genus Homo. And then following anamensis, we've got from about 3.8, 3.6 million years ago, we've got Australopithecus afarensis. Australopithecus afarensis is very well studied, particularly because there's been an abundance of material found about afarensis, and one famous fossil dubbed Lucy that is almost complete. We also have another fossil of afarensis that is referred to as Lucy's baby, but precedes Lucy by about 0.3 million years in geological time, so clearly couldn't be Lucy's baby, but is the fossilized remains of an almost complete toddler about the age of 3. What we notice with afarensis is that the teeth, or at least the front teeth, are still fairly ape-like, particularly the canines. The canines are shearing. We see parallel tooth rows that are more similar to chimpanzees than what we see eventually in modern humans. Long arms with ape-like wrists, not risks, that are still well suited to climbing, but the fingers are shortening, which is indicative of spending less time in the trees. We also see a large degree of sexual dimorphism in body size, on the scale that's more similar towards gorillas versus that of chimpanzees and modern humans. Afarensis is still primitive enough, particularly with its dental development, and its relatively small brain size, that it's a strong candidate for an ancestor for all later hominins. And so this is of particular importance. We also see a lot of variability. We're not going to have these later very specialized Australopiths as ancestors of the genus Homo. We're going to see a pretty early divergence between Australopiths and the genus Homo that dates to about 2 million years ago. And so, yeah, just speaking to the primitive nature of Afarensis. Afarensis is also, we've got a very important find that are what we call the Laetoli footprints. So found at a field site named Laetoli. And these are fossilized impressions of footprints of about 6 Australopiths that were striding across the grassland. And so we can see with that the big toe is fully in line with the rest of the toes. So we've got this commitment towards the stability granted by that loss of the grasping big toe. We don't yet see an arch in the foot that's going to come later. And the gait or the stride length is shorter than what we're going to see with later hominins. However, we've got this full commitment to bipedality. So Anamensis was out in the grasslands with a fairly large home range size, striding bipedality. Or Afarensis, I'm sorry. The later Australopiths show a degree of separation between what we call gracile and robust Australopiths. As Australopiths persisted, they became more specialized, they became more derived as they adapted to a variety of ecological niches. And so the biggest difference between gracile and robust Australopiths has to do with the overall robustness of the skull and this commitment to really vegetarianism. Um, and so in robust Australopiths, those that are in the genus Paranthropus, we've got a further commitment towards thick, fibrous, tough vegetation. This leads to some changes in the skulls, an overall increase in the body size, which is associated with an increase in the skull size. We see the largest teeth of all Australopiths, particularly the molars and the premolars. They've got incredibly heavy enamel, all of which is adapted for this diet that's based on hard, rough vegetable foods. So, that mythical sweet potato, for example, they are fully committed to a diet based on these mythical hard sweet potatoes. They've got lower, deeper jaws that give more areas for muscle attachment. They've got pronounced attachment sites, including the sagittal crest and the brow ridges for their chewing muscles. They have broad cheeks. The cheeks are the area where the masseter muscles attach. So all of this robusticity of the skull is associated with providing surface area that these chewing muscles can attach to. So their chewing burden was astronomical. They have a small cranial capacity. We find Paranthropus aethiopicus at about 440 cubic centimeters, Paranthropus boisei, Paranthropus robustus at about 510 to 530 cubic centimeters. This isn't statistically significantly different. This really reflects that overall greater robusticity of their skull altogether and their overall slightly larger body size than the gracile Australopiths. So these are like gorillas, really. I mean, not in terms of being predecessors of gorillas, but when we look at the skeletal material of gorillas versus chimpanzees, the gorillas are much more massive, and that massive body size, those massive features of the skull are associated with a vegetarian diet, specifically thick, hard, fibrous vegetable material. The gracile Australopiths are kind of lower profile, a little more petite. They are small brain, roughly about 440 cubic centimeters. This isn't, again, statistically significantly different. It's just associated with an overall a smaller body size. They are large-toothed relative to kind of proto-human ancestors, relative to Sahelanthropus, Auroran, and Ardipithecus, but not nearly as extreme as Paranthropus. So the Australopiths as a whole trend towards larger, thicker teeth with thicker enamel. But we see a specialization between Paranthropus and Australopithecus. And so we've got Australopithecus africanus included in this group. We also have Australopithecus sediba included in this group. Australopithecus sediba is a relatively recent find. The first specimens were found in about 2009 in South Africa. Lee Berger, who we'll talk about later with Homo naledi as well, was out in the field. His son Matthew actually found one of the first fossils of Australopithecus sediba. He was out playing with his dog and ended up tripping over something and landing kind of he was out with his dog playing fetch and ended up tripping over something and landing basically face to face with some fossil skeletal material. Australopithecus sediba represents a unique mix of primitive Australopith traits, including the smaller brain size, including um, teeth that aren't really diminutive, these large teeth, including a heavier pronounced brow ridge, etc., as well as derived homo traits. Some of those derived homo traits include a more rounding of the brain case and such. So they've got a small brain, long arms, curved fingers, but their fingers are short, not long, indicating that even though they could climb and their shoulders are still indicative of climbing, they weren't climbing quite as frequently. They've got a very flexible lower spine that is more in line with the curvature of spines of the genus Homo. And through changes in the brain case and the impressions left in the brain case, some indicators of possible brain reorganization. So they're occupying a more complex dietary niche. And specifically, these gracile australopiths, instead of focusing on these thick, fibrous, tough vegetable materials, instead, we're starting to take or partake in the consumption of meat. And so specifically, and through their association with stone tools, they're starting to take advantage of scavenging opportunities. They're not fully hunting. So it is at this late point between gracile australopiths and early members of the genus Homo that we start to consider meat eating part of the suite of behaviors that hominins demonstrate. Early hominins weren't eating meat. They were certainly going after what we call extracted foods or foods that are embedded in substrate. But these were predominantly going to be things buried in the ground. These were going to be these roots and tubers, not very much game. And so, some of the ways that we see some of the things that we see with respect to the consumption of meat. I mean, we find stone tools, Luna. We find stone tools, we find hominin fossils, and we find what we call faunal assemblages. That is, the bones of animals that would have provided meat. Now, we can use a number of characteristics to determine whether we're talking about scavenging or active hunting. For example, we look at the association of tooth marks and cut marks. If the tooth marks are laid down on the bone first and the cut marks then follow, then the hominins were scavenging these materials. They were brought down and chewed on by a predator first. If the cut marks are laid first and the tooth marks overlay the cut marks, then hominins were actively hunting these animals, and then scavengers were getting access to their kills when the hominins were done. Um, Another factor that we can look at is the utility of the faunal element. What that means is what bones of prey items are found. And when groups are scavenging versus hunting, we find a lot of what's called head and hoof. We're seeing metatarsals, metacarpals, fingers and toes, basically. And when you think about your fingers and toes, there's not a whole lot of meat on them. You can get into marrow, but still you're not even seeing large bones that are really thick that are going to have a lot of marrow inside of them. So not much meat available on the hoof, not much meat available on the head. You do have, of course, the brain. That can be pretty valuable, but that's not what we're seeing. It's not they're not zombies. It's not a specialization on brain consumption. So the meat that they're getting are going to be things like the cheek meat, which are the masseter muscles of the animals that they eat, as well as perhaps the tongue, etc. So again, not high yield in terms of meat available. When we see active hunting, we see hominins associated with the femur. And the femur, the thigh, the rump, I mean, these are the areas of animals that have the biggest kind of bang for your buck, the most meat available. So around 2 million years ago, maybe a little bit preceding this. We've got a shift from these early Australopiths into both the evolutionary pathway that gives rise to these specialized Australopiths, the gracile and the robust Australopiths. But we also see around that same time a different side branch, the origins of the genus Homo. So, a little bit more than 2 million years ago, things get really interesting. We talked about how these later Australopiths, the gracile and robust ones, weren't really able to, are very, very specialized for particular dietary niches. And so they're not likely ancestors to the genus Homo, but those early ones, particularly Australopithecus afarensis, is a likely candidate. And so sometime a little more than about 2 million years ago, we see the divergence of these later Australopiths and the genus Homo. The genus Homo is marked predominantly by significant increases in brain size. And so specifically, we've got two fossils that both date to about 1.8 million years. Homo habilis, who was found in the Olduvai Gorge, with a brain case of about 631 cubic centimeters, larger front teeth than Australopiths, narrower premolars, and then an even larger brain, more recent Homo, Homo possibly named Homo rudolfensis, who dates to between 1.8 to 1.4 million years ago. This fossil was found in East Turkana and demonstrates a brain size of around 775 cubic centimeters. There's a little disagreement as to whether we should consider Homo habilis and Homo rudolfensis two separate species, particularly because we'd already seen greater sexual dimorphism in Australopiths than was initially expected, and the idea that perhaps these could just be regional variations in kind of polytypic variants of one species. But Homo rudolfensis does demonstrate this larger brain case and larger teeth. Importantly, this demonstrates that we've got multiple species living in the same region at the same time, with gracile Australopiths, robust Australopiths, and potentially two members of the genus Homo. So remember when we were talking about evolutionary theory, when we were talking about Darwin's finches, if we're going to have multiple species occupying the same time and space, they're going to do so by specializing on different ecological niches. And so we've got the robust Australopiths eating this thick, fibrous, tough vegetation. We've got gracile Australopiths with a mostly plant-based diet and a little bit of scavenging. We potentially have these two members of the genus Homo doing considerably more scavenging, occupying this slightly different ecological niche. So we will move forward from this point with our next module as we start talking about Homo erectus. We're going to see further increases in brain size that come with the emergence of Homo erectus. We're going to also see this transition to active hunting, and so we'll characterize Homo erectus as the world's first hunter-gatherer, and Homo erectus leaves Africa as a hunter-gatherer and really sets the stage for an ever-increasing importance on cultural means of adaptation. So before we wrap up, we're going to talk about these basic processes of paleoanthropology. The first step in practicing paleoanthropology is to select and survey a site. The second step is to excavate your sites and recover your fossils. The third is to designate individual finds with specimen numbers for clear reference. So, for example, we've got KNMER with a certain number afterwards. This allows materials to kind of have this universal designation so that it's easy to find them again when you so desire. You then have to clean, prepare, study, and describe the fossils. Sometimes this can take upwards of a decade. You've got to compare these with other fossil material, particularly in this ecological or chronological time frame if you can. That, of course, requires having accurate dates for your fossils. You also have to compare fossil variation within this known range of closely related, within known ranges of variation of closely related both living primates and ancestral or fossil primates. So we're trying to place or get a clear picture of how they relate to and compare in terms of these ancestral and derived characteristics. We see taxonomic names assigned then to fossil material, whether they designate or allow us to designate a new genus, new species, etc. And then you've got to publish your material in peer-reviewed sources. This is how you get the science out there for everyone to be able to review and analyze. This is what kind of bears up to further scrutiny. And so we essentially end here with this kind of tentative phylogeny of early hominins. So we've got these early specimens, Sahelanthropus chacadensis and Auroran tugenensis. We've got Ardipithecus that is established with a fair degree of certainty, perhaps, that it's ancestral. I mean, we still see question marks in the relatedness here. We don't draw lines of relatedness to between Sahelanthropus and Auroran tugenensis to either Ardipithecus or these early Australopiths. We do see though this potential for Ardipithecus to be ancestral to both. We don't know how Anamensis fits within this early kind of picture. We don't know if Anamensis is ancestral to Afarensis or if Anamensis and Afarensis independently evolved from Ardipithecus. Anamensis is potentially ancestral to Kenyanthropus, the skull that is associated with these Lomekwian tools, potentially. We do know that Afarensis is likely ancestral to later forms of derived Australopiths, including the gracile Africanus garhiancetaba and the robust Ethiopicus boisei and robustus. And Afarensis is presumed to be ancestral to the genus Homo. Now, why do we have question marks here? It's because any new fossil find can change the picture of how all of these species fit together. And so we've got to recognize that new finds that haven't been made yet, reanalyses of finds that have been made, aren't they have the potential to reshape how we draw our tree of evolutionary relatedness. Something was going on, though, ecologically between about 4 and 1 million years ago that led to a large degree of variability. It was the spread of the savanna. It was anthropoids in the form of hominins moving out into the savanna. And it was this period of a lot of evolutionary tinkering, a lot of kind of fitting together of ancestral and derived traits. So we had a wealth of variability in early hominins, most of which really leads to evolutionary dead ends. As we talk about the adaptive patterns of early African hominins, we note that all early hominins had restricted ranges. Each hominin species exploited a relatively small area that could have easily then been led to isolated populations. These isolated or geographically isolated populations result in a higher degree or a higher perhaps effect of genetic drift that could lead to rapid genetic divergence and eventual speciation. Most of these species appear to be at least partially tied to arboreal habitats. And so we see this retention all the way through Homo habilis of the ability to climb trees. And so this is particularly key in this scope of of protection, right? You're going to climb trees at night because you're going to be particularly vulnerable at night. You're also going to be, you're going to climb trees um, when you're threatened. That threat could be due to predators, that threat could be due to conspecifics, neighboring groups, etc. So each hominin species exploited a relatively small area. Then we see rapid changes that result from natural selection and genetic drift. Most of these species appear to be except for some of these early members of the genus Homo, there's very little in the way of an evolutionary trend of increased body size or greater encephalization. We see brain sizes that are pretty consistent and spot on with what chimps were experiencing. So this may be related then to a lack thereof of associated patterns of stone tool manufacture. If these populations were using tools, they were Oldowan pebble tools with the possibility of Lemequian tools, but they were relatively unspecialized tools. They also all show an accelerated pattern of growth and development, as evidenced by dental eruptions. That is, there's not much of a childhood, really, because they don't have much of a brain change from um, these chimpanzees. So if the brain hasn't changed very much, then we also aren't going to see corresponding changes in the pattern of development. Um, they were nursing for probably about 5 years, and then they were self-feeding from about 5 years on. This doesn't mean there wasn't some cooperation with these later Australopiths. There would have been some food sharing and cooperative defense of these scavenged opportunities, but it wasn't nearly as significant as it's going to become later in our development. So this increased encephalization is going to be associated with childhood emerging with Homo erectus. It's going to be associated with longer periods of parental provisioning. It's going to be associated with a larger degree of food sharing and kind of cooperative endeavors in gaining access to food. So, in summary, be able to identify when the earliest primates appear and what kind of conditions fostered the appearance of early primates. Be able to recognize that most went extinct. We've had a wealth of primate diversity that resulted in evolutionary dead ends, that only a few were ancestral to modern extant primates. The Eocene is this age of prosimians, the first appearance of anthropoids. The Oligocene is this age of anthropoids, first appearance of hominoids. The Miocene is this age of hominoids, first appearance of hominins. Later time periods, the Pleistocene and Pliocene, become the ages of hominids or hominins. Accurate dating is imperative for understanding the primate and hominin evolution. The two common methods are relative and absolute. Be able to identify the key features of early hominins and how these early hominins vary. Be able to pinpoint perhaps our first hominins with Sahelanthropus chuckedensis and Ararontugenensis, and recognize that hominins are only found in Africa for the next 5 million years, and then be able to characterize hominins as pre-Australopith, ancestral Australopith, derived Australopith, and early Homo, and be able to identify kind of how they fit together evolutionarily.